Hey, good morning. Welcome back to part 2 of the Central Dogma. So we're going to pick up right where we left off. So we left off with an introduction to the genetic code. We talked about how does a cell read codons and how does it know which amino acid is needed for the protein. So today what we're going to do is we are going to get into the molecular mechanisms. And specifically we're going to talk about part 2, and that is transcription. So we're going to talk about how is DNA copied to RNA? How does RNA polymerase use DNA as a template, and how does it make RNA? How does it know where to start? How does it know where to end? And then we're going to talk a little bit about something called RNA processing, which is a process that eukaryotes do, but prokaryotes do not. So we will get into that. And again, our overarching question that we are working on answering is, if proteins are responsible for nearly everything that occurs in a cell, how do we make proteins? How do we take our set of instructions and how do we take that DNA and use it to eventually get to protein? So we're going to start with transcription. We've seen this flow of genetic information, so the idea that DNA is copied to RNA. So we're really focusing here on the first half of this gene flow. So what do we need for transcription? And normally we would do a brainstorm activity here, but because we are online, I'm going to go ahead and jot down some things, but I encourage you to pause the video before I do that and see if you come up with the same things that I do. So first and foremost, of course, we need our template. So we're going to need our DNA template. That's our set of instructions. That's what we're going to copy. We need the thing that's going to do the copying. So we are reading DNA and making RNA. So we are going to need RNA polymerase. This is going to be our enzyme that does this process. And of course, we need the building blocks. And those building blocks are going to be our A's, U's, C's, and G's. So these are all the pieces that we're going to need. And of course, we could add other things like, well, we're building, so we're going to need energy as well so that we can actually do the polymerization of the nucleic acid. But we're going to talk mostly about the pieces themselves. So just like we have rules for DNA replication, we have rules for making RNA, and they're very, very similar. So if you recall, for DNA replication, we said that replication starts at a specific site. Well, it turns out that transcription is also going to start at a specific site. And that site is called a promoter. In DNA replication, that site was called an origin of replication. Here in transcription, we call it a promoter. It's going to promote the RNA polymerase to start transcribing or copying the DNA into RNA. And our template, which in transcription is DNA, is read from 3' to 5'. If you recall from DNA replication, DNA is also read from a 3' to a 5' direction. And the next rule is actually going to be very similar as well. So RNA is synthesized in a 5' to 3' fashion. And the reason for that is RNA polymerase, just like DNA polymerase, can only add to the 3' end. So just like we need a free 3' OH, or that free 3' end for DNA replication, the same is true for transcription. So again, many similarities between transcription and replication. The key difference is that in DNA replication, the goal is to just copy the DNA. That is all, and it is only used when we are ready to undergo mitosis and meiosis in that S phase of the cell cycle. And we use a DNA polymerase because we read DNA and make DNA. In transcription, This can happen at any point of the cell cycle, and the goal is to, usually at the very end, to have a protein made. And so in transcription, we are reading DNA and making RNA. Because we are making RNA, we are using an RNA polymerase. So let's do a brief overview of transcription. And transcription is going to have 3 steps, if you will. It's going to have a step of initiation. And so this is figuring out where to make the mRNA. Right? So where do we start? We're not just going to pick a random spot on the DNA. There are specific sequences. The next step is elongation. And so this is building the RNA, and this is going to be using the NTPs, so the ribonucleotide triphosphates. And then last but not least, unlike DNA replication where we copy the entire genome, for transcription, we only copy the set of instructions that we need. So again, if your friend is letting you borrow their notes, you're not going to rewrite their entire notebook from the semester. You are only going to rewrite the notes that you missed. And so we have to have a signal of some sort that signifies termination, and this is going to be place to stop the RNA polymerase. So before we get into all of these steps, let's talk a little bit about the structure of DNA for transcription. So of course, DNA is our template, so we're going to draw what that looks like. DNA is double-stranded. It's, of course, anti-parallel. So it is also complementary. We've got hydrogen bonds holding it together. All right, so here is our DNA. And we're going to have a couple of pieces in this DNA. We're going to have this blue region here. And this blue region is our promoter. And a promoter is a special sequence, and really it's a special DNA sequence, and this is going to tell RNA polymerase where to start making mRNA. And promoters have different DNA sequences. There are specific promoters that are for specific genes. Usually, not always, many promoters have A's and T's, and for the same reason that origins of replication have A's and T's. Now, after we have the promoter, we have kind of the next big chunk, and this is referred to as the gene. And the gene itself, these are the instructions to make the protein. And so this is eventually, if we were to take that DNA and change the T's for U's, this is where those codons come from. This is where those instructions on which amino acids we add in come from. So those are the actual specific instructions. So we have DNA sequence before those instructions that tells us where to start, and it's basically like a, hey, RNA polymerase, there's a gene coming up, you might want to start here, so you make sure you copy the whole thing. That's kind of how you can think of a promoter. Now, of course, at the very end of that gene, we're going to have this pink sequence, and this pink sequence is the terminator sequence. And again, this is a special sequence in DNA, and this tells the RNA polymerase to stop making mRNA. And so we call this entire thing oftentimes is referred to as a transcriptional unit. And so we have a promoter, a gene, and a terminator sequence. And much of our genome is just repeats of this. So after that pink box, which is a terminator sequence, there might be a promoter sequence for another gene. And then we'll have another gene, another terminator sequence, and that repeats over and over again. And so this is the structure of one transcriptional unit, if you will. All right, so we talked about the 3 steps. So we've got initiation, elongation, and termination. Now let's actually go ahead and let's draw the process out. So we're going to start on the left here, and we're going to draw our DNA. And for our DNA, we are going to have, I'm going to label it here as P as a promoter, and then somewhere at the end, we're going to have our terminator sequence. So the first thing that is going to happen is RNA polymerase is going to find the promoter. So let's go ahead and draw an opened up DNA here. And when I say that RNA polymerase finds the promoter, it's a tad bit misleading because there are proteins called transcription factors that help with this. But for the sake of this class and for the sake of simplicity, we're just going to refer to this as RNA pol doing a lot of the work. So RNA polymerase begins at the promoter. It's going to open the DNA. So for DNA replication, we have a helicase that opens the DNA. It turns out RNA polymerase is really special. It has helicase activity, which means it can open up DNA. So it's going to open the DNA, and then it's going to begin to read the DNA, and of course, it's going to make RNA. So we're going to kind of continue this process up here. So over time, depending on the gene length, this can take a couple hours to a couple minutes. We'll see that that RNA polymerase has moved down the DNA. So now here's our promoter. Our RNA polymerase is down here. It's getting near that terminator. And we have made some RNA, and this is going to continue until that terminator sequence is reached. When that terminator sequence is reached, RNA polymerase falls off, and at the end of transcription, our result is we end up with the DNA that we started. So that DNA does not change. We also have that RNA polymerase, and we also have a single-stranded RNA that is our mRNA. So this is our messenger RNA. This is our copy. All right, so we've drawn our mRNA as 5' to 3'. And the reason that we've done that is, if you recall, all nucleic acids are synthesized in the 5' to 3' fashion. And anytime we think about synthesis, we also have to think about what is the template that is used. So what we're going to do is we're going to do a practice problem. So here we have on the top, we have a strand of DNA. This is our template strand. And our template strand is shown here in the 5' to 3' direction. And based on what you know, if our start site, so here in the middle is our promoter, if this is our start site, will transcription go to the left, which is A, or will it go to the right, which is B? So go ahead and pause the video, and as a hint, if you get stuck, recall what those rules are for nucleic acid synthesis. All right, hopefully you have paused the video, and we're going to walk through this problem together because this is asking us to put a lot of pieces together. So there's a couple key points that we have to remember. Number one, all nucleic acid synthesis is templated. So that means that we have to have a nucleic acid that we read. And whenever we read nucleic acid, whether it is DNA for replication or DNA for transcription, we always read nucleic acid 3' to 5'. Why? Because we can only synthesize 5' to 3' because of our polymerases. And so DNA and RNA polymerases synthesize nucleic acids in the same direction, and they read in the same direction. So the other thing that we have to remember is everything is anti-parallel. So if everything is anti-parallel, we can actually figure out for this molecule where the 5' and 3' should be. So here we have our DNA. Our DNA is written in a 5' to 3' direction. So that means that whatever molecule is made has to be made 5' to 3' and must be anti-parallel, which means that this is our 3' end and this is our 5' end, because everything is anti-parallel and everything is based on hydrogen bonding. So if we take a look at B, this means that this would be the 5' end and this would be the 3' end. Again, all based on this idea of anti-parallelism. Well, can a polymerase add to a 5' end? No, right? There is no way that B is possible. Now let's take a look at A. So because this is our 5' end of DNA here, this will be the 3' end and this will be the 5' end. Can we synthesize and add to a 3' end? Heck yeah, right? That's a polymerase's favorite end. That's the only end they can add things to. So that means that for this RNA polymerase, the way that this is drawn, it's going to move towards A. And this brings up a very, very important point, and that is, what is the template strand? So we said everything, whether it's replication or transcription, requires a template strand. So how do we figure out for DNA what the template strand is? And that's what we're going to do next. And this takes us to something known as coding and template strands. And I'm going to write some of our rules up here just to remind ourselves. And that is the idea that polymerases read 3' to 5' and they synthesize 5' to 3'. So keep that in mind as we are working through this. So I'm going to come up with a sequence here. Let's do ATG, ATC, TTC, GCA. And then now that we have that, we can go ahead and we can complement. This will be our DNA. So we have TAC, TAG, AAG, and CGT. So this is our DNA. And now what we have to do is we have to figure out, well, which of these strands is going to be used to make mRNA, because our mRNA can only be made in a 5' to 3' passion. So if we take a look at this, we can actually start to see, well, if we use this strand down here, this is in a 3' to 5' fashion, and you and I know what we should be looking for. So for mRNAs, we always look for a start codon, and a start codon is AUG, and in DNA, that's ATG. So we actually have one of those here, but we can't use this strand because this is in a 5' to 3' direction. So we're going to use this strand down here, and I'm going to make my mRNA orange here. So if we start to write this now, T base pairs with A base pairs with U, C base pairs with G, and then T, so A, U, C, U, C, G, C, A. So here is our mRNA now that we have synthesized. So we followed all the rules. We read in a 3' to 5' fashion, and we synthesized in a 5' to 3' fashion. So our mRNA is 5' to 3', which is good because that's how our codon table is set up. So now we can actually start to give these things some names. So this bottom DNA strand right here, this is the one that we read. And so anytime we have a strand that is read by the RNA polymerase, we call this the template strand. And the reason we call it the template strand is because that is what the polymerase uses as a template to synthesize the mRNA. Now, when we've been talking about the codon table, we've been saying that the genetic code is really taking DNA and changing the T's out with U's so that we can then use those to figure out what the codons are. Well, let's take a look here really quick at our mRNA. Isn't this mRNA exactly this top strand of DNA, except taking those T's and switching them out with U's? So we've got AUG, ATG here. We have AUC, ATC, so on and so forth. So this top strand is identical to the mRNA. And so we call this the coding strand. And the coding strand just means that this looks like the mRNA, except of course those T's are switched out for U's. So the coding strand, again, is the same sequence as mRNA, except it has T instead of U. So another way that we can think about this is actually looking at the bubble that we have drawn here. So we've got our transcription drawn here in the bottom right-hand corner. And let's go ahead and actually draw a transcription bubble here, but with the example that we've been talking about on this page. All right, so what we're going to do is we're going to write 5' to 3' here, 5' to 3' here. And let's go ahead and add our bases in here. All right, so there is our top strand drawn exactly the same as our top strand above. And then let's draw our bottom strand as well. All right, so now we have both of our strands drawn here. And I think this is hopefully going to really help us see what is the template and what is the coding strand and how this mRNA will be synthesized. So we'll keep our colors consistent here. So we can actually take a look at our ATG here. So here is our start, and the start of HERS is for translation, but we look for it in DNA. And so that means for our mRNA, we would have an AUG. So whenever you find the start, or what will be the start codon in mRNA, that actually automatically tells you that this is the coding strand, which tells you that the bottom strand has to be the template strand. And we can further figure that out based on the directionality. So if we take a look here at our 3' end and our 5' end, here is our ATG. But we can't read 5 to 3 because we would then have to synthesize 3 to 5, which we can't do. So that means we're going to start right here at this 3' end, and we're going to lay a 5' end there, and we're going to start replicating our mRNA here, which gives us the same sequence that we got up here. So the key point here that I hope has come across is that DNA and RNA, regardless of the type of nucleic acid you are both synthesized in an anti-parallel and complementary fashion. All right, so this is a really difficult topic. This is really hard, and this is something that I struggled with as a student. So what we're going to do is we're going to do some practice with this. So I encourage you to pause the video, and with this strand of DNA here, so this is DNA, there's a couple things I want you all to do. So first, I want you all to determine the complementary strand of DNA, to find where the start codon would be. So recall that our start codon is from 5' to 3', an AUG in RNA, and so that means in DNA it's got to be ATG. And then determine the mRNA and determine the amino acid. So this will give you practice with that codon table as well. So hopefully you have paused the video, and what we're going to do is we are going to walk through this together. So I've just put this on a blank page just so that I have more room, because y'all know I write really big. So let's start with our DNA sequence, and let's start by complementing it. So we know that DNA is anti-parallel, so this will be the 3' end. A base pairs with T, T with A, so we've got TAC, CGC, GAA, ATC, and GTA. So here is our DNA. All right, now what we have to do is we have to look for the start. And the start in DNA is ATG in a 5' to 3' fashion. So I'm going to go ahead and look for one, and it turns out if we start on this top strand here, we have an ATG right here. It's in the 5' to 3' orientation, so it is in the correct direction. So we're going to go ahead and we're going to circle that, and our gene goes to the right. So here's our ATG. That would be our first codon, so AUG. So let's go ahead, and because we found our ATG, we automatically know that this is our coding strand. And so our mRNA should look like that top strand, which means our bottom strand is our template strand. So let's go ahead and figure out the mRNA. Now, when it comes to determining the mRNA, there's two ways you can do it. You can do it by knowing that this is the coding strand, and knowing that the coding strand is the mRNA-like strand, and all we have to do is replace the T's with U's. Or we know that's the coding strand, which makes the bottom strand the template strand, so we can use the template strand, and we can actually use our rules of anti-parallelism and complementation. So I'm going to do it that way. So we're going to start with a 5', since this is a 3' end. This TAC, when RNA polymerase reads this, it's going to put an AUG. For the next triplet here, we have CGC, so we'll have GCG. The next triplet will have CUU. The next one will have UAG. And last but not least, we'll have CAU. Because this is a 5' end, we will end with a 3' end. So here's our mRNA. Now, using the codon table, we can actually go ahead and translate this. So AUG is MET, GCG is ALA, CUU is LEU, and then UAG is one of our stop codons. And so we're going to stop there. That CAU would technically not be translated, so we're going to leave that one alone. All right, so most of you have probably gotten this far on the problem, but there's a key point here. And the reason that I like to give this practice problem is because genes can go in both directions. And what I mean by that is, if you think about it, if we have billions of base pairs, You know, it seems a waste to just have genes going always one direction on DNA, especially when we have two strands. Why not have genes going to the left and to the right, right? We just need a promoter, a gene, and a terminator. So let's take a closer look at this bottom strand here. So at this bottom strand, again, we always want to look for an ATG in a 5 to 3' fashion. So let's start at the 5' end. And this takes a little bit of work and a little bit of skill, and it has taken me years to be able to do this and to spot these easily. And if we read from right to left backwards, we actually have A, T, G. Wait a minute, it's in a 5 to 3' direction. Isn't that what we're looking for? So it turns out we actually have two genes. The second one here is going to go to the left. So we're going to do gene number 2, and we're going to figure out its mRNA. So now, using this lighter green color here, here's our ATG. And for when we find an ATG, we know that that makes it the coding strand. So for gene number 2, this is the coding strand. And that means that the top strand has to be the template strand. So for our second mRNA, we're actually going to use this top strand as the template. So here's our 3' end, which means anti-parallel. So mRNA is going to start with 5'. We have TAC, which is going to be AUG. And then GAT, when that polymerase reads that, it's going to put in CUA. When the polymerase reads TTC, because of complementation, we will have AAG. Next, we'll have GCG, CAU. And then we'll reach our 3' end. And then our start codon, which is MET, LEU, LYS, ARG, and then HIS. So as you can see, we have one piece of DNA, but two genes going in different directions that are coding for different proteins. So I realize that this is challenging, and I know that this is difficult. So there are actually a bunch of practice problems that are up on Moodle, and there is an answer key that is posted as well. And the practice problems are going to encompass transcription and translation, and we're actually going to build in the next video as well. So rest assured, lots of practice problems are coming your way, whether it's via the lecture or via Moodle. So now let's talk about RNA processing. So RNA processing is a really cool concept, and the key here is that this only happens in eukaryotes. So this does not happen in bacteria or archaea, and part of the reason for this is it ties back to the structure of cells. And for RNA processing, I like to think of RNA processing as taking something called pre-mRNA to mature mRNA. And in our prokaryotic friends, after transcription, mRNA is ready to go. So they don't mess around with any of these steps. This only happens in eukaryotes. So in eukaryotes, when we make mRNA, it's actually technically called pre-mRNA. So it's not a mature form of messenger RNA yet. There's a couple things that we have to do to get it to the mature form. So the first thing that we're going to do is we're going to take this 5' to 3' pre-mRNA, if you will, so immature mRNA. And the first thing we're going to do is we are going to add a cap. And when we add a cap, I'm going to make my cap a circle here. So here's our 5' to 3' end. This is called a methyl G cap. And the reason it's called a methyl G cap is because if we take a look at the zoomed in image here for this cap, it has a methyl group here on what should be a guanosine. So we've got our methyl G cap, and if you're wondering why the heck do we add a cap, the whole goal with our cap is to protect against degradation, and the cap is actually a signal also for translation. So after we add a cap, there's a second thing that we do. So now we have a pre-mRNA with a cap. The second thing that we're going to do is we're going to add a poly-A tail. And a poly-A tail is exactly what it sounds like. We're adding a bunch of A's at the end. And the goal of this, or why we do this, is to protect against degradation. So now we have protected our 5' end with a cap, so no nucleases can chew that up. We've protected the 3' end with a tail, so again, no nucleases can chew that up. And the other thing that the tail is important for is it's actually important for export out of the nucleus. So there are 3 things we have to do for RNA processing. Number 1, add a cap. Number 2, add a poly-A tail. And the third thing that we have to do, we are going to demonstrate by thinking about the dystrophin gene. So we're actually going to talk a lot about muscular dystrophy in this class because we're going to do an entire case study on this. So dystrophin is one of the coolest genes, and part of the reason it's so amazing is that it is the largest gene that has been currently characterized. So it has 2.4 million base pairs, which is absolutely and utterly amazing. And the dystrophin gene is one that is found to be mutated in the many forms that we have of muscular dystrophy. All right, so let's do a practice problem here with this dystrophin gene. So we just said that the DMD, or the dystrophin gene, has 2.4 million base pairs. All right, well, dystrophin, which is the protein, so this is one of the proteins that you can get, has 3,685 amino acids. So because we know the length of the protein, we know how many amino acids it has, we can actually figure out the number of nucleotides that are needed to encode this amino acid. So if we take 3,685 and we multiply it by 3, right? Because each amino acid is encoded by a codon, which is a triplet of nucleotides. So we take that 3,685, multiply it by 3, we get 11,055 nucleotides, which means B. But wait a minute, that doesn't add up, does it? How the heck do we get 11,000, a little over 11,000 nucleotides, but the gene's 2.4 million base pairs? What is going on there? There has to be something else going on. And in fact, there is. And this brings us to the third thing that has to happen for RNA processing. And the third thing that has to occur is something called splicing. And splicing has to be one of the coolest things that happens. It's certainly one of my favorite things to talk about. We're not going to get into all of the molecular mechanisms of it. I talk a lot about it in cell biology, but it's just such an amazing process. So the way that I think about splicing is the way that this cartoon shows it. So here we've got different pieces of our mRNA, and some of the pieces are saying here, some are saying absent, some are saying I'm stuck, and this gets back to what occurs in splicing. So let's go ahead and draw out what our mRNA looks like so far. So we've only been drawing our mRNA with one color, and what I'm going to do is I'm actually going to draw our mRNA with black and blue ink. So here we go. This is what we have done so far in our pre-mRNA to mature RNA processing. We have added a cap and we've added a tail. Now we are on step number 3, which is splicing. And we have 2 things going on here. We've got the black ink, which is going to represent are exons, and I like to think of exons as expressed. So these are the parts of the mRNA that will be left in the mature mRNA, and those are the parts that are expressed. Now we have these blue parts. These blue parts are referred to as intervening sequences, so they intervene between the exons, and these are the sequences that are called introns. So introns comes from intervening sequences, and these sequences are cut out. So they are not expressed, and there is an entire group of hundreds of proteins, and it is called the spliceosome. So I told y'all, we love in science to add "ome" to everything. It just makes it sound sexier, right? Spliceosome sounds so much better than splicing machinery. But anywho, there is this entire complex of hundreds of proteins that is called the spliceosome, and essentially it works like molecular scissors. So these are supposed to be scissors, pardon my horrible drawing. And what happens is in splicing is we cut out the introns and we paste together the exons. And so at the very end, after we've capped it, after we've added a tail, and after we have spliced it, our end product is finally an mRNA. Let's label these exons as 1, 2, and 3, with exons 1, exons 2, exons 3, and then our poly A tail. So here is our mature mRNA. So eukaryotes have a lot of steps to take before they can get to the dang translation part. We certainly are very, very complicated. And if you're wondering, okay, why the heck do we do splicing? How did this evolve? One of the reasons that we have behind the idea of splicing and why we think it evolved is you can actually mix and match exons. So here we have drawn an mRNA that has exons 1, 2, and 3. But even though exons are considered expressed, you can sometimes actually cut exons out as well. So this same mRNA could have yielded a protein that only had exon 1, or a protein that only had exon 2, or a protein that only had exon 3, or you could mix and match any of the exons that are there. And so what this allows us to do is to have one gene, but have many different proteins that come from it. Now, as you can imagine, things get very complicated very quickly. So for a gene like the dystrophin gene, where it is 2.4 million base pairs long, we do a lot of splicing, and sometimes mistakes happen in splicing that can lead to mutations down the line. So splicing is a really complex process, and for the sake of this class, we are going to leave it as cutting out the introns and pasting together the exons. So let's do a quick practice problem to make sure that we are on the same page. So for a mature eukaryotic mRNA, which of the following best represents what it will look like? So go ahead and pause the video and walk through this practice problem. So hopefully you have walked through this, and when we have a mature mRNA, we're going to have a 5' end, which we have our cap, and then we're going to have our coding sequence. And first, within our coding sequence, we need to have a start. So somewhere we're going to have an AUG, and we're going to write stop because there's 3 stop codons. So somewhere we're going to have a stop, and then we're going to have a poly A tail. So this is what a mature eukaryotic mRNA looks like. And if we take a look at our choices, we can cross off anything that doesn't start with a cap, and we can cross off anything that has extra pieces. So we can cross off E. And if we take a look at B and D, they're both actually saying the same thing. The only difference is that B here says that this is the poly A signal, so B or D could be correct. Clearly, Dr. Hubbs did not prove this problem enough before she put together the lecture, but for both B and D, again, they're saying the same thing. In one case, we just have the poly A tail. In another, we have the poly A tail, and I told you that it was a poly A signal. So we can just pretend that I did that on purpose. I was just testing you, right? Anywho, I digress. Let me know in the comments below if you have any questions on anything in transcription. So we have made it to the process where we now have a mature eukaryotic mRNA for our bacterial and archaeal friends. All they had to do was go through transcription. They didn't have to go through any of this RNA processing. So when we come back in part 3 of this video, we're going to talk about how do we take our mature mRNAs, whether it's eukaryotic or prokaryotic, and how do we translate those? So thanks for hanging out with me in part 2, and I will see you in part 3. Bye, everyone.